Hi everyone, my name is Tanjimei. Today I'm going to present my final year project with the title of Distributions, Abundance and Substrate Preference of Giant Clam at Pulau Tioman, Pahang and Batu Rakit Lighthouse, Terengganu, Malaysia. So first of all, giant clam is the biggest bivalve in the world. They are important in terms of ecology, food source and aquarium trade. However, they are found declining in the wild due to human exploitations, marine cultures and pollutions. The problem that we found there are no updates of giant clam status in Pulau Tioman since 1998, no published data for giant clam and Batu Rakit Lighthouse, and very limited info regarding substrate preference of giant clam in Malaysia. So, this study is to update the current status and provide baseline data in order to have better understanding and help in conservation and management program on giant clam. The objective of the study is to determine the distributions and abundance of giant clam at Pulau Tioman and Batu Rakit Lighthouse, and also to study the substrate preference of giant clam according to species at both study areas. The methods that we use are the combination of belt transect method and photochromic method, which are responsible for the study of distributions and abundance and substrate preference respectively. So, there are 3 transit lakes at Batu Rakit Lighthouse and 9 at Pulau Tioman. And then, after the transit lake, we take the depth where the giant clam found and also measure the shell length. The picture of giant clam with quadrant were taken. And then, we identify the giant clam species and sort the data according to species, size, and depth. We also identify the substrate type and calculate the benthic cover using CPC 4.1 software. For the data analysis part, we analyze the distributions and abundance of giant clam through species composition density, and also size and depth distributions according to the criteria at the left. Well, how to know whether the data is significant or not? We run the crucible test and Mann-Whitney test at 95% significant level through SPSS software. For substrate preference, we also consider the embeddedness and the habitat preference of giant clam according to species. So, there are 2 species of giant clam at Batu Rakit Lighthouse and also 3 in Pulau Tioman, which is Tridana maxima, Tridana squamosa, and also Tridana crocea. So we can see that Tridana maxima recorded the highest density among the study areas, which means Tridana maxima was the most abundant species found. However, when we run the crucible test to see the difference on giant clam density between 2 study areas, the results show was not significant for all species available. For size distributions, Tridana squamosa was larger than Tridana maxima and Tridana crocea. For depth distribution, Tridana squamosa was found distribute deeper than 2 other species, and it is significant. For substrate preference, Tridana maxima was mostly found in coral reef that rich in massive corals, and they are partially embedded on dead coral and leaf massive corals. The difference between substrate preference of Tridana maxima at Pulau Tioman and Batu Rakit Lighthouse was not significant. And same, the correlations between substrate type and degree of embeddedness of Tridana maxima was not significant too. For squamosa, it was normally found in coral reef that rich in branching corals and they are freestanding on coral rubbles or sandy substrata, where the difference between substrate preference of Tridana squamosa between 2 study areas was significant. However, the correlations between substrate type and the degree of embeddedness of Tridana squamosa was not significant. For crocea, it was normally found fully embedded on dead coral with algae, and because they have special organs to secrete acid that allowing them to penetrating the calcium-based substrata. The correlation between substrate type and degree of embeddedness of Tridana crocea was not significant too. For conclusions, Tridana maxima was the most abundant species found at both study areas, where Tridana squamosa was larger than 2 other species, and it was found distributed deeper compared to Tridana maxima and Tridana crocea. For substrate preference, Tridana maxima preferred dead coral with algae and massive corals, whereas squamosa preferred sandy and coral rubble. Tridana crocea mostly found fully embedded on dead coral with algae. And by the end, we hope this study may help in conservation and management program on giant clam. These are the references that I use. That's all from me. Thank you.